Have you ever wondered how complex the world of chromosomes can be, especially when they multiply beyond the norm? Welcome to the intriguing world of polyploidy. It's a state of having more than 2 complete sets of chromosomes. Sounds unusual, right? But it's surprisingly common across many species and plays a vital role in evolution and the formation of new species. But that's not all. Polyploidy also has a darker side. It contributes significantly to tumor heterogeneity, the diversity of cells within a tumor, making it a tough adversary in our battle against cancer. It also plays a role in drug resistance, making certain treatments less effective. So, is polyploidy a fascinating anomaly of nature or a key player in evolution and disease? Well, it's a bit of both. As we delve deeper into the mystery of polyploidy, we'll unravel its complexities and reveal its true nature. Let's delve deeper. How does polyploidy occur, you ask? Recent research has some interesting answers. Polyploidy is not a random phenomenon. It's engineered by a variety of mechanisms that all lead to an abnormal increase in the number of chromosomes. Let's start with cytokinesis failure. This is the process that occurs at the end of cell division, where the cell splits into 2 daughter cells. However, when this process fails, it leads to the creation of a multinucleated cell. This cell then undergoes a process called endoreplication, which results in polyploidization. Essentially, the cell duplicates its DNA without dividing, leading to more than 2 complete sets of chromosomes. Another mechanism that drives polyploidy is mitotic slippage. Mitosis, as you might remember from biology class, is the process by which a cell divides its chromosomes into 2 identical sets. But sometimes, things don't go as planned. Mitotic slippage happens when chromosomes fail to segregate properly during cell division, leading to the creation of tetraploid cells, cells with double the usual number of chromosomes. Finally, let's talk about endoreplication. This is another process where the cell duplicates its DNA but does not divide. It's a bit like cytokinesis failure but works in a slightly different way. The result, however, is the same: cells with an abnormal number of chromosomes. But there's more to the story. Defects in DNA damage response pathways also promote polyploidy. You see, these pathways are like the cell's quality control system. They ensure that cells with damaged DNA do not proceed to cell division. However, when these pathways are defective, cells with damaged DNA can slip past these checkpoints, leading to polyploidy. These mechanisms illuminate how cells can end up with more than their fair share of chromosomes. Each mechanism is like a piece of a puzzle, helping us understand the complex nature of polyploidy. And with each new discovery, we move one step closer to understanding how polyploidy contributes to evolution, development, and disease. Polyploidy doesn't just occur, it triggers a cascade of effects that alter cellular function. Once polyploidy takes hold, it's akin to tossing a pebble into a pond. The ripples spread far and wide, reaching every corner of the cellular landscape. The first ripple touches gene expression. When cells have more than 2 complete sets of chromosomes, their genetic material alters significantly. This affects the expression of genes, leading to changes in the production of proteins and other gene products. The cell's identity, its functionality, and even its destiny can be rewritten by this shift in gene expression. Next, the ripple reaches cell proliferation and differentiation. Polyploidy can influence how quickly cells divide and the types of cells they become. This can result in phenotypic differences or observable traits that distinguish polyploid cells from their diploid counterparts. The polyploid cells may proliferate more rapidly or differentiate into cell types that are unexpected, adding another layer of complexity to their behavior. The ripple effect doesn't stop there. It disrupts cell cycle progression and checkpoint control, causing cells to divide without proper checks and balances. This disruption can lead to genomic instability, making the cell's DNA more prone to damage and mutation. This instability is a double-edged sword. While it can drive evolution and adaptation, it also makes cells more susceptible to diseases, including cancer. Finally, polyploidy plays a key role in driving tumor evolution and resistance to therapy. By generating genetic diversity, polyploidy allows tumors to adapt and evolve in response to environmental pressures such as chemotherapy. This is one of the reasons why some cancers become resistant to treatment over time. Polyploidy, therefore, is not a silent player, but a significant influencer in the cellular world. Its ripple effects touch every aspect of cellular function, from gene expression and cell proliferation to genomic stability and disease progression. Understanding these ripple effects is crucial to unraveling the complexities of polyploidy and its role in health and disease. With knowledge comes power. Understanding polyploidy opens doors to potential therapeutic avenues. The complexity of polyploidy has presented researchers with a wealth of opportunities for therapeutic intervention. Given our understanding of the mechanisms and consequences of polyploidy, we can now look at exploiting these for potential treatments. A key strategy here is to target regulators of polyploidy. You see, polyploidy doesn't happen in a vacuum. It's driven by regulators such as cell cycle checkpoints and mitotic kinases. By selectively targeting these regulators, we could potentially eliminate polyploid cells, disrupting the irregularities they cause. But there's another side to polyploidy. Its induction often leads to genomic instability, which, while causing a host of problems, also presents us with a unique opportunity. This instability creates vulnerabilities that we can exploit for therapeutic purposes. By targeting these weak spots, we could develop new strategies for treating diseases where polyploidy plays a significant role, such as cancer. However, our therapeutic arsenal doesn't stop there. With the advent of genome editing technologies, we are now in a position to correct polyploidies at their source. By editing the genome, we can restore chromosomal stability, reversing the effects of polyploidy and putting us one step ahead in our fight against diseases caused by chromosomal abnormalities. As we delve deeper into the world of polyploidy, we are continually finding new ways to turn this complex phenomenon to our advantage. Each discovery, each insight brings us closer to innovative therapeutic approaches that could transform the way we treat diseases. As we continue to unravel the complexity of polyploidy, we edge closer to innovative therapeutic approaches that could transform disease treatment.